Hi, I'm Kathleen Hupfeld. I'm a PhD student in Dr. Rachel Seidler's lab at the University of Florida. And today I'll be speaking about a part of our ongoing work investigating brain and behavioral changes with spaceflight. And specifically, my talk will focus on our work using functional MRI to characterize neural vestibular changes with spaceflight. So here's just a brief outline of my talk today. I'll start with discussing background information and the broader goals of our group's neuromapping study. First, I'll touch on several of the big picture motivating factors for developing and conducting our neuromapping study. We know that spaceflight negatively affects various aspects of human performance. For instance, post-flight, astronauts experience transient problems with walking and balance, as well as difficulties with fine motor coordination. This is a publicly available video, and not one related specifically to our study, showing an astronaut soon after their return to Earth from an ISS mission. As you can see, they're experiencing pretty pronounced walking difficulties. In addition to such behavioral data in humans, various rodent work also suggests that brain structure changes with spaceflight. And finally, our work with head-down tilt bed rest, a spaceflight analog, further supports that spaceflight factors result in extensive brain and behavioral changes. Thus, these points led to the two overarching aims for our neuromapping study. First, we aim to understand how spaceflight affects brain structure and function, as well as the time course of such effects. And second, we aim to characterize how such brain changes with spaceflight relate to performance changes to better understand the functional significance of brain changes with flight. Since we began our neuromapping project almost 10 years ago, there's been accumulating evidence from our group and others supporting that spaceflight affects human brain structure. Several of these effects of spaceflight on the brain include a pre to post-flight upward shift of the brain within the skull. Demonstrating this is a video from our recent paper. And other effects include headward fluid shifts, ventricular expansion and increases in total CSF volume, and declines in white matter integrity and connectivity. But despite this work on brain structure, less work to date has tested how spaceflight affects human brain function. So one of the main goals of the neuromapping project has been to study how brain function changes with spaceflight. So the remainder of my talk will focus on how spaceflight influences human brain function, and specifically vestibular function. So here I'll briefly review why spaceflight particularly influences the vestibular system. One component of the vestibular system, the otoliths, sends linear accelerations, including head orientation relative to the gravitational vector. While on board the ISS, when somebody's exposed to microgravity, otolith signaling is altered. And consequently, the central nervous system, or the CNS, must adapt in order to properly orient the body. Then, after return to Earth, we know that astronauts experience these transient difficulties performing vestibularly mediated behaviors like walking, balance, and visual orientation tasks. And these difficulties extend beyond peripheral declines like muscle weakness, and are thought to be due in part to in-flight adaptive central vestibular changes and to sensory reweighting, or the central nervous system downweighting the altered vestibular inputs and upweighting other afferent sensory signals like visual and somatosensory signals. So this was the goal of our present work, to use functional MRI to study how central processing of vestibular information might change with spaceflight and relate to behavioral performance. So now I'll discuss further details regarding our methods and hypotheses. Our neuromapping study as a whole included 15 astronauts who completed approximately 6-month-long ISS missions, and several of these individuals were female. Participants completed 6 testing sessions in total, each of which included brain imaging, or MRI, and behavioral testing. And 2 of these sessions occurred before the ISS mission, and 4 of these sessions occurred after the ISS mission. I will briefly note, astronauts also completed some testing on their first day back, as well as in-flight cognitive measures. However, in this talk, I'll focus only on the data collected at the 6 blue time points, because these indicate the times at which we collected both MRI and behavioral data. So at each of those 6 indicated time points, we collected a battery of MRI measures. And here we'll focus on the vestibular functional MRI measure. So to collect this measure, we used an MRI-compatible pneumatic tapper, shown in the top row images here. The tapper stimulated both the left and right lateral cheekbones for several minutes each, alternating between blocks of tapping and rest, while we collected functional MRI data in an MRI scanner. We've previously demonstrated that this pneumatic tapper method successfully targets the vestibular system through bone conduction, because it induces ocular vestibular evoked myogenic potentials, or OVEMs, that we can record from the eye muscles. So in response to this vestibular stimulation, we typically see activation, or upweighting, shown in red, of vestibular inputs, which include the bilateral insula, and specifically the retroinsular cortex and the right parietal opercular area 2, or right OP2 region. And at the same time, we also see widespread deactivation or suppression, shown in blue, of cross-modal sensory regions, including portions of the somatosensory motor and occipital cortices. So to restate this once more, typically in response to vestibular stimulation, the neural response that we see includes upweighting of vestibular inputs and concurrent downweighting of sensory areas while the brain is processing these vestibular inputs. In addition to characterizing brain activity during vestibular stimulation at each of the 6 time points, we also examined possible relationships between vestibular brain changes with spaceflight and functional performance. We focused on 4 behavioral tests: the functional mobility test, which is an obstacle course designed to require maneuvers similar to those performed during egress from a space vehicle after landing; the sensory organization test 5, SOT5, and the SOT5 with head movement. In these tests, participants stand as still as possible on a sway reference space with their eyes closed. And in the head movement condition, participants make head pitch motions synchronized to a metronome tone. And finally, the rod and frame orientation test, which assesses the extent to which participants rely on a visual frame of reference to perceive Earth's gravitational vertical. We also collected a number of other sensory motor and cognitive measures at these time points and in flight, although we won't discuss those in greater detail here. We had 2 primary hypotheses regarding our vestibular fMRI data. First, we tested for the time course of brain changes with spaceflight. We tested for regions that showed a stable baseline at the 2 pre-flight time points, followed by either increases or decreases with flight, and then recovered to baseline levels by the last post-flight time point. We predicted that somatosensory and visual regions would follow the orange line pattern here, showing post-flight decreases in the suppression response that we see during vestibular stimulation. And second, we predicted that pre to post-flight changes in brain activity during vestibular stimulation would associate with pre to post-flight changes in performance on vestibularly mediated behaviors, including walking, balance, and visual orientation. So now we'll move on to our vestibular brain and behavioral results. First, our analysis examining the time course of brain changes during vestibular stimulation yielded widespread pre to post-flight changes in brain activity, followed by a return to baseline patterns by 6 months post-flight. So specifically, after spaceflight, multiple sensory motor, temporal, and occipital regions showed less of the normal suppression response that these areas usually show during vestibular stimulation. So that is, post-flight, these regions exhibited less of that blue suppression response to vestibular stimulation that I depicted earlier in my talk. So this plot shows an example of one of the resulting regions, the left precentral gyrus. The group average extracted brain activity values from this region are plotted, and the hypothesized contrast weights are shown by the dotted orange line. So all of the regions overlaid on the left followed our hypothesized contrast pattern, showing a stable baseline, followed by reductions in suppression with spaceflight, and then subsequent recovery towards baseline values by our last time point. So next, we examine how these brain changes with spaceflight correlated with behavioral changes. However, I'll just briefly illustrate the changes that we found in behavioral performance among these astronauts. Of the 4 vestibularly mediated behaviors we focused on, 2 of these changed significantly with spaceflight when examining the 6 time points that corresponded best to our MRI scans. Performance on the FMT obstacle course slowed significantly from pre to post-flight. So here, times on the x-axis and FMT performances on the y-axis, and higher y-axis scores indicate slower completion time. And similarly, equilibrium quotient scores on the SOT5 task significantly declined from pre to post-flight. So here again, time is on the x-axis and SOT performance is on the y-axis, and lower y-axis scores are worse. So given these pre to post-flight declines in FMT and SOT5 scores, we then tested whether pre to post-flight brain changes correlated with pre to post-flight behavioral changes to help unpack the functional consequences of the pre to post-flight decreases in suppression that we saw in our longitudinal model. We didn't find any associations with FMT, but we did find that pre to post-flight changes in the visual cortices correlated with pre to post-flight changes in SOT5 balance scores. So more specifically, greater reductions in suppression of these visual areas from pre to post-flight correlated with better post-flight balance. So here on the x-axis, we have pre to post-flight change in SOT5 performance, and further to the left indicates worse post-flight balance, and further to the right indicates better post-flight balance. And on the y-axis, we have pre to post-flight change in brain activity during vestibular stimulation from one of the visual cortex clusters shown on the left. So higher y-axis values indicate greater reductions in suppression from pre to post-flight. And as a third exploratory aim of this work, we tested how vestibular brain changes with spaceflight correlated with mission duration. This analysis yielded a relationship in which shorter flight durations correlated with greater pre to post-flight reductions in cerebellar suppression. So again, this reduction in suppression response is similar to that overall longitudinal effect we found throughout the somatosensory and visual cortices. So here's a plot depicting these results, although to maintain privacy, we don't show the individual data points. On the x-axis is flight duration, and again on the y-axis we have pre to post-flight change in brain activity during vestibular stimulation from the right lobule 5 cluster shown on the left. And here higher y-axis values indicate greater reductions in suppression from pre to post-flight. And so next I'll talk through our working interpretations of these findings. First, we'll consider the time course result in which we found pre to post-flight reductions in suppression of visual and somatosensory regions. So that is, we found pre to post-flight reductions in the normal suppression or downweighting response that we see with vestibular stimulation. We think that this result suggests in-flight adaptive neuroplasticity and sensory reweighting. So that is, while on board the ISS, astronauts encounter novel afferent vestibular signals due to the altered gravitational environment. And while in flight, this results in sensory reweighting, or downweighting of vestibular inputs, and upweighting of inputs from other sensory processing regions, such as the somatosensory and visual cortices, to process vestibular information and properly orient the body. Then, post-flight, some of this sensory reweighting remains, and this is evident as post-flight reductions and suppression of the somatosensory and visual cortices. And we see this decreased suppression soon after landing, but this change then recovers towards baseline values in the months after return to Earth. And this sensory reweighting interpretation fits with the accumulating body of in-flight EEG work and animal work, in addition to one of our recent head-down tilt bed rest papers from the VAPOR campaign, in which we also found decreased suppression of multiple brain regions during vestibular stimulation across the bed rest intervention. And secondly, we examined how these brain changes with flight related to behavioral performance changes. And examining behavioral performance changes is incredibly useful in interpreting the functional significance of brain changes. For more details on this, you can check out our recent review paper in Neuroscience and Biobehavioral Reviews, where we discuss the importance of leveraging behavioral data to understand brain changes in the context of spaceflight. So in the present work, we found that greater pre to post-flight reductions in suppression of the visual cortices associated with less pre to post-flight balance declines. And we suggest that this again fits with the idea of in-flight compensatory sensory reweighting. And so specifically while in flight, there's increased reliance on an upweighting of the visual system for processing vestibular information and maintaining body orientation. And then after return to Earth, those who showed more efficient upweighting of the visual system to process vestibular information were also the best performers at the standing balance task when readapting to normal vestibular inputs on Earth. And finally, we found that shorter flight durations correlated with greater pre to post-flight reductions in cerebellar suppression. So interestingly, this is similar to a result from one of our past studies examining brain structure. In that paper, we found a relationship between mission duration and cerebellar white matter microstructure. So similar to here, shorter mission duration correlated with greater pre to post-flight decreases in the white matter microstructure measure. And while we hesitate to overinterpret these results, we suggest that both our past and present cerebellar findings reflect cerebellar adaptation over time. So that is, there may be some initial cerebellar changes that take place earlier in flight, and these are followed within mission adaptations. And finally, I'll note several conclusions based on these findings and our planned future directions for this work. First, our findings suggest that spaceflight results in adaptive neuroplasticity and sensory reweighting for vestibular processing. And second, these identified changes in neural vestibular processing with spaceflight impact post-flight balance performance. And finally, as a whole, these findings provide greater scientific understanding of neural compensation and adaptation to perturbations to vestibular function. And going forward, we're in the process of completing analyses of our other functional and structural MRI metrics with spaceflight. And big picture, we plan to compare our results across our spaceflight and bed rest data sets, which include perturbations like elevated CO2 and countermeasures like artificial gravity. And for more information on these ongoing projects, you can check out the poster presentations from several of the other folks in our lab. And that concludes my presentation. I would like to acknowledge all of our co-authors and collaborators who've been working very hard for many years now on our neuromapping project, as well as all of the astronauts who participated in this work and our multiple funding sources. And during the live session, I'll be happy to hear any feedback and take any questions on this work. And I've also put my contact information on the slide if anyone would like to follow up with comments in that manner. Thank you.